huge diversity of organisms they are present over the earth's surface. Out of these various plant, animal as well as other organisms, they are really important in various fields like agriculture, forestry, industry. It means once we have gathered the information about these organisms, this information should be appropriately stored so that this information can be utilized for the future purpose, for the future reference. Now, biologists, what they have done? Biologists, they have standardized certain techniques, certain procedures which helps in storage of all this information, and all these techniques and procedures together going to constitute taxonomic aids. So you got what are the taxonomic aids? They are techniques, procedures and stored information which is utilized for the classification and identification of organism. So let's write the definition for the same. Taxonomic aids, they are techniques, procedures as well as stored information used for classification and identification of organism. Right? So all this information should be properly stored so that we can utilize this information in the future for the future reference. Now here I am going to describe few taxonomic aids like flora, manual, monograph, journal, catalog, herbarium, botanical gardens, museum, zoological parks, zoos and taxonomic keys. So these different taxonomic keys I am going to describe here. Let's start with the first 5. We are going to do comparative account of this, comparative study of these. Yes, the first one, flora. Flora is a book which gives the information of different types of plants present in a particular area. Means it is going to give the information about listing of plants in an area. So what is flora? It is a book which provides information about listing of plants in a defined geographical area. It will give listing of plants, but along with that it will also give the information about habitat of plant. Habitat, that is the environment where the plant is surviving. So along with the listing, it is also give the information about habitat, environment of a plant. So here we can cite one example. Example is Flora of Delhi. Flora of Delhi. It was written by the scientist Meke Maheshwari. Yes. So I am citing example here, that is Flora of Delhi. Listing of all the plants which are present in Delhi region. This was performed by the scientist J.K. Maheshwari. Right? Next one now. Next one is manual. Manual is also going to provide a listing of plants in a particular area. Listing of plants in a particular area, and along with that it is also going to give the description about the plant. So manual is going to provide the listing of plants. Along with that, it is also going to describe characteristic features of plant. And one more additional thing. It gives information about specific group of plants, like manual of cultivated plants, manual of aquatic plants, manual of medicinal plants. So what is the additional feature? It gives information about specific group of plants. Specific group of plants. Yes, so I am citing one example here, that is manual of aquatic plants. Got this? Now after the manual, now next taxonomic aid, that is monograph. Yes. What do you mean by mono? Mono means single. So it gives information about a single group. It gives information about a single taxon. Yes. It gives information. It gives information, detailed information about a single group, single taxon. Now one example which I can cite here, that is monograph of the genus Avena. Monograph, the genus Avena. Yes. Avena, it is the scientific name for oat. Yes, Avena, I can say generic name for the oat. Right? Nowadays, what is happening? Now, scientists, they are working on this Avena and they are trying to increase its nutritional status. So, scientists, they are working on this plant, they are working on this genus and trying to increase the nutritional status of the plant. Yes, so it means this is all what? This is all current research, ongoing research. Ongoing research, the results of the ongoing research is published in a taxonomic aid, and that taxonomic aid is known as journal. Yes, please write it down. Journal. Yes. Journal will have the results of ongoing research. Journal will have the information of current research. So it means any new discoveries, any current research, it is going to be published in a taxonomic aid. That aid is journal. Let's write one example here. One example of the journal is Flora. Yes. Now after the journal, now the last category, that is catalog. Catalog. Catalog also gives a listing of organism. Yes, listing of species, and generally this listing is done in a alphabetical manner. Yes, alphabetical arrangement of species. Along with that, it is also going to give the brief description. Right? Alphabetical manner arrangement. Alphabetical arrangement of species, and along with that, also going to provide description of different characteristics about the species. Yes. So these 5 taxonomic aids, they are very much clear to all of you here. Now we are going to deal with the next one, that is herbarium. So now we are going to deal with next taxonomic aid, that is herbarium. Herbarium is a place जहां पर हम dried pressed plant specimen को preserve करते हैं. Yes, so it is a place where dried pressed plant specimen, they are preserved, which are mounted over the sheets. Yes, they are stitched, they are pasted over the sheets. So let's write it down. Herbarium is a place for storage of dried, pressed plant specimen mounted on herbarium sheets. So we can easily say herbarium is a storehouse of plant. Yes, it is a storehouse of dried pressed plant. यहाँ पर हमने प्लांट को सिर्फ प्लेस नहीं करना है, यहाँ पर सिर्फ प्लांट्स को डंप नहीं करना है. These herbarium specimens, they are also going to be systematically arranged according to the most widely accepted classification system. Yes, so these specimens, they are arranged systematically according to the widely accepted classification system. And in most of the herbaria, the classification system which is utilized, that is Bentham and Hooker system. So arrangement of specimen is based upon Bentham and Hooker. Most accepted one, classification system. Right? Now first of all, we are going to see now how this herbarium specimen can be prepared. अब इस herbarium specimen को बनाना कैसे है, कौन कौन से steps इस specimen को बनाने के लिए utilize किए जाएंगे. Steps required for the formation of herbarium specimen. First one, collection, drying, poisoning, mounting, stitching, labeling, and deposition. So these different steps we are going to discuss here. First of all, we are going to start with collection. collection. जिस प्लांट को हमने कलेक्ट करना है, if that plant is a herbaceous plant, it is a small herbaceous plant, that in this case whole of the plant is removed. Yes, whole of the plant is collected along with the root system. The plant which is collected, if it is herbaceous, that in this case whole plant is collected along with the root system. जिस प्लांट को अब हमने कलेक्ट करना है, अगर ये वुडी है, it is a tree or shrub. If it is a woody plant, tree or shrub. If it is a tree or shrub, so in this case only part of plant is utilized, twig of plant is utilized. So in this case only twig of plant is collected. Twig showing the features like leaves, fruits and flowers of the plant. Now the size of the twig should be from 30 to 40 centimeter. प्लांट को हमने अब कलेक्ट कर लिया है. अब इस प्लांट को हमने डाल देना है पॉलिथीन बैग के अंदर और एक मेटालिक बॉक्स के अंदर, जिसको हम कहते हैं वैस्कुलम, ताकि इस प्लांट का डिटोरेशन ना हो एंड टू प्रिवेंट द लॉस ऑफ मॉइस्चर फ्रॉम द प्लांट. तो जो प्लांट आपने कलेक्ट किया है, इस प्लांट को आपने डाल देना है पॉलिथीन बैग के अंदर या एक मेटालिक बॉक्स के अंदर. Yes, what this metallic box is known as? This is known as vasculum. Yes, why all this is done? This is done to prevent the deterioration of the plant and to prevent loss of water from it. Now, after the collection, now the next step, that is drying. अब इस प्लांट स्पेसिमेन को हमने ड्राई करना है, तो हम क्या करेंगे, इस प्लांट स्पेसिमेन को न्यूज़पेपर के ऊपर स्प्रेड कर देंगे. ईदर वी कैन यूटिलाइज न्यूज़पेपर और ब्लॉटिंग पेपर, बिकॉज बोथ न्यूज़पेपर एंड ब्लॉटिंग पेपर दे कैन एब्सॉर्ब द मॉइस्चर एंड लेट द स्पेसिमेन ड्राई. यस, so now we are going to spread the specimen over the newspaper or blotting paper. Specimen plant material is spreaded over newspaper or blotting paper. अब इन न्यूज़पेपर्स को हमने रख देना है फोल्डर के अंदर एंड दिस फोल्डर इज प्लेस्ड इनसाइड अ साइंटिफिक टूल that is known as field press. So now these newspapers, they are placed inside a folder. This folder is placed inside a machine that is known as scientific tool that is known as field press. Now field press, what it is going to do? It is going to perform uniform pressing of specimen. Uniform pressing. It means along with the drying also the uniform pressing of the specimen is done. Now these newspapers, when they will absorb the moisture, these newspapers, they will become wet, moistened. So these newspapers which are moistened, they should be repeatedly changed. Otherwise there can be pathogen attack. Okay, so the moistened newspapers are changed repeatedly. Yes, this process is repeated again and again till the specimen becomes completely dry. Yes, till the specimen is dried. Specimen is pressed, specimen is dried. Now it is the time for next step, that is poisoning. स्पेसिमेन के ऊपर हम केमिकल स्प्रे करेंगे ताकि हम इस स्पेसिमेन को बचा सके फ्रॉम द अटैक ऑफ पैथोजन. यस, सो पॉइजनिंग केमिकल्स, they are utilized to protect the specimen from attack of pathogen. Let's see now which chemicals they are used. One is 0.1% mercuric chloride. Mercuric chloride, it has the ability to kill fungi. So you can say it will protect the specimen from fungal attack. Or this is fungicide. Got this? Along with that, DDT is also utilized. Yes, it has the ability to kill the insects. So, it is insecticide. स्पेसिमेन प्रेस हो गया, ड्राई हो गया एंड आपने उस स्पेसिमेन को पॉइजन भी कर दिया. अब ये टाइम है कि अब हम इस स्पेसिमेन को पेस्ट करेंगे ऑन अ हर्बेरियम शीट. सो नाउ इट्स द टाइम फॉर mounting, stitching and labeling. Right? Specimen is placed over a herbarium sheet. Herbarium sheet. International size of the herbarium sheet is 41 into 29 centimeters. International size is 41 into 29 centimeters. Let's say this is herbarium sheet. Yes, this is herbarium sheet. Over the herbarium sheet, now we are going to place the specimen. Yes, we will place dried pressed specimen. This specimen is pasted over here. It is stitched over here. Now specimen को आपने यहाँ पर stitch कर दिया है. Now we are going to also label the specimen. Yes, can you tell where the labeling of the specimen is performed? Labeling is performed on the right hand side, on the lowermost corner, that is here. Here the labeling is performed. What should be there in the labeling? In the labeling there should be scientific name, scientific name of the plant. Along with that there should be vernacular name. Vernacular name, family to which this plant belongs. Family, then name of collector, date and place of collection. Along with that, we can also write some of the economic importance, that is the utility of plant. Right? Specimen is now totally ready. It is mounted over the herbarium sheet. Now the last step, now we are going to deposit this specimen, that is deposition. Last step is deposition. Now dried pressed specimen, it is deposited in the herbarium where it is systematically arranged. Deposited in herbarium where it is arranged. Yes, how it is arranged? It is arranged according to the accepted classification system, and the one which is most widely utilized, that is the Bentham and Hooker system. So the different steps of making herbarium specimen is very much clear to all of you. So now we are going to cite examples as well as uses of herbarium. Let's write certain examples. RBG, that is Royal Botanical Garden. Royal Botanical Garden के साथ एक herbarium associated है. Yes, Royal Botanical Garden, it is in Kew, England. और इसके साथ जो हर्बेरियम एसोसिएटेड है वहां पर प्रेजेंट है मैक्सिमम नंबर ऑफ स्पेसिमेन, दैट इज ओवर 6 million. Next one, CNH, that is Central National Herbarium. This is in Calcutta, India. Now the number of specimens which are present in this Central National Herbarium, that is around 2 million. Now what are the uses of herbarium? One, the herbarium, they help in accurate identification of plant species. Herbarium, they help in identification of plant species. Suppose I have got one specimen from a locality and I want to know which specimen it is. So what I will do? I will study the characteristic of the specimen which I have collected, and I will compare these characteristics with the herbarium specimen. And this comparison can quickly tell us what is the identity of the plant which I have collected. So by performing the comparison with herbarium specimen, I can easily perform identification of plant. So this is one of its utility. You can also say it is used for alpha taxonomic रिसर्च. अगर तुम्हारे सामने एक हर्बेरियम स्पेसिमेन पड़ा है तो उस स्पेसिमेन को देखकर आप प्लांट के कौन से फीचर बता सकते हैं. यू कैन टेल द मॉर्फोलॉजिकल फीचर्स ऑफ द प्लांट, एक्सटर्नल फीचर्स ऑफ द प्लांट, दे कैन बी इजिली स्टडीड, and you know the taxonomy which deals with morphological feature. That taxonomy is known as alpha taxonomy. Got this? So with the help of herbarium specimen, morphological features, they can be studied. Morphological features can be studied, and taxonomy which deals with morphological characteristics, this is known as alpha taxonomy. Got this? So it means we can easily say here that herbarium, they are quick referral system for identification or quick referral system for taxonomic studies. Quick referral system for identification for taxonomic studies. After the herbarium, now we are going to deal with the botanical gardens.